to everybody. Duchenne muscular dystrophy is an X-linked disease that affects 1 in 3,500 live male births. The onset is around childhood, leading to a necessity of a wheelchair around 10 years old and to a premature death for cardiac and respiratory failure. The cause of the pathology is a mutation in the DMD gene, the gene that encodes for the dystrophin. Dystrophin is a cytoskeletal protein that anchors the sarcolemma to the actin cytoskeleton. With the C-terminal domain, dystrophin interacts with the protein of the dystrophin glycoprotein complex. DMD gene is the biggest gene of the human genome. Thus, a lot of different mutations might occur in this gene. In Duchenne muscular dystrophy, all of these mutations determine the complete absence of dystrophin. The loss of this protein compromises the integrity of the dystrophin glycoprotein complex, causing the rupture of the sarcolemma during each muscle contraction. In dystrophic patients, the persistent degeneration of the myofibers determine a condition of chronic inflammation and the depletion of muscle satellite cell pool, with consequent replacement of the damaged myofibers with adipose and fibrous tissue. Despite the huge effort to find a cure to treat DMD, to date, the standard treatments are glucocorticoids with their limited efficacy and lots of side effects. Sertoli cells are the major component of seminiferous tubules in testes. Sertoli cells release a lot of trophic and immunomodulatory factor, creating an immune-privileged microenvironment that protect germ cells from the immune system attack. Thanks to the factor that Sertoli cells release, they survive longer in case of engraftment and protect allo and xenogenic engraftment of organs and tissues. So, during the years, free or macroencapsulated Sertoli cells have been tested in several experimental animal models of different diseases, such as type 1 or 2 diabetes, Huntington disease, Laron syndrome. In our laboratory, thanks to a collaboration with the group of Professor Giovanni Luca and Riccardo Calafiore, Sertoli cells have been isolated from prepubertal pig testes and macroencapsulated in barimalginate microcapsules. Macroencapsulated Sertoli cells have been injected in the peritoneal cavity of MDX mouse, the most used animal model to study DMD, in the acute phase of the pathology. And after 3 weeks from a single injection, in Sertoli cells treated mice, we observed a reduction of inflammation and necrosis and adipose and fibrous tissues in the muscle, thanks to the release of trophic and anti-inflammatory factors from Sertoli cells. But in Sertoli cells treated muscle mice, we observed also an upregulation of eutrophin expression, the paralog of dystrophin, and this was due to the release of irregular beta-1 by Sertoli cells. All these positive effects translate into an improvement of muscle performance in MDX mice treated with Sertoli cells compared to MDX mice treated with empty microcapsules. To go on this investigation as a part of a larger project funded by the Duchenne Parent Project, we decided to study the effect of Sertoli cells on myoblasts. Co-culturing Sertoli cells with C2C12 or human healthy and dystrophic myoblasts, or using a medium conditioned by Sertoli cells. So, first of all, we observed that in murine C2C12 myoblasts, the presence of Sertoli cells after 24 hours of differentiation determine an increment of the number of myoblasts, as we can see by phase contrast microscopy, and as we have confirmed by MTTSA and BRDU positive cells. But we observed also that Sertoli cells determine a reduction of apoptosis and a reduction of oxidative stress. Similar results were obtained in human healthy and dystrophic myoblasts. Also in this case, we observed an increment of the number of myoblasts in presence of Sertoli cells, and we confirmed by cell count. Moreover, cytofluorimetric analysis at the same time point show that switching myoblasts from grow medium to differentiation medium determine a reduction of the percentage of cells in the S phase, whereas in presence of Sertoli cells, the percentage of cells in the S phase were higher, with values similar to myoblasts in grow medium. To have more information about the role of Sertoli cells in the differentiation process, we analyzed the expression of myogenic markers, in particular myogenin and type 2 myosin heavy chain. As we can see by Western blotting analysis, in human myoblasts, the presence of Sertoli cells delay the expression of myogenic marker in the first part of the differentiation process, whereas in dystrophic myoblast, at 6 days of differentiation, in presence of Sertoli cells, we have an upregulation of the expression of myogenin and type 2 myosin heavy chain. And these results was confirmed also by real-time PCR analysis for the gene of myosin heavy chain in dystrophic myoblast. So this data suggests that Sertoli cells favor the terminal differentiation in dystrophic myoblast. The first part of the result suggests that Sertoli cells or Sertoli cells-derived factor exert a promyogenic activity, but this promyogenic activity of Sertoli cells do not alter the myogenic potential. Indeed, we observe that in both murine CTC12 and human dystrophic myoblast, at 72 hours of differentiation, in presence of Sertoli cells, we have an increment of the number of cells, but this increment was accompanied by an increment of the fusion index and of the number of nuclei inside each myotubes. So, the promyogenic activity of Sertoli cells do not impair the formation of myotubes. Moreover, since it has already been demonstrated that in a primary culture of myoblast, it's observed a transdifferentiation from myoblast to myofibroblast, we analyzed the expression of fibrogenic markers. And as we can see by real-time PCR analysis, in dystrophic myoblast at 6 days of differentiation, the presence of Sertoli cells determine a reduction of the expression of fibrogenic markers, in particular COL1A1, fibronectin, and CTGF. Moreover, we analyzed the effects of Sertoli cells on preformed myotubes. And as we can see, on human healthy and dystrophic myotubes, Sertoli cells determine an upregulation of eutrophin expression independently from the mutation, as we can see by Western blotting analysis and immunofluorescent staining. As we have already demonstrated for CTC12 and MDX primary myotubes, also in human dystrophic myotubes, the upregulation of eutrophin expression induced by Sertoli cells follow an irregular beta-1 ERK1/2 pathway, as we can see by this Western blotting analysis recently performed using an irregular beta-1 blocking antibody or an inhibitor of the tyrosine kinase ERB-B2 receptor family. So we might conclude that Sertoli cells or Sertoli cells-derived factors stimulate in murine CTC12 and human healthy and dystrophic myoblast the cell proliferation in the first part of the myogenic process without alter the myogenic potential. Moreover, Sertoli cells in dystrophic myoblast stimulate the terminal differentiation and reduce the expression of fibrogenic markers. So this data suggests that a Sertoli cells-based approach might be useful in the early phase of the muscle regeneration to improve the number of cells necessary to replace the damaged muscle mass. Moreover, Sertoli cells in human healthy and dystrophic myotubes determine an upregulation of utrophin expression in an irregular beta-1, ERK1/2 dependent manner. Altogether, these data support that macroencapsulated Sertoli cells or Sertoli cells-derived factors might be a potential treatment for Duchenne muscular dystrophy. Thanks to all for the attention, and I would like to thank my Professor Guglielmo Sorci and especially my colleague Sara Chiappalupi.
Thank you very much, Laura, for this very, very interesting talk. I think there are some questions.
Is your turn, Capucin.
Yes, thank you for this very nice presentation. I was wondering, have you done the secretome of these cells? Are you able to know exactly what they are secreting, or is it ongoing? Or
this is a beautiful question and also frequent question. Unfortunately, Sertoli cells are a biofactory. They secrete a lot of trophic, anti-inflammatory, and immunomodulatory factor. It's not easy to understand if it's 1, 2, or 3 of them that make all these positive effects, or maybe it's what we think is that the altogether of this factor might be useful to have these positive effects.
Okay. And if I may follow, I have 2 more questions. I was wondering, have you tested their effect on fibroblasts? Because you showed that they have a reduced expression of profibrogenic on myoblasts, but have you looked directly on the non-myogenic cells, what effect they have?
Okay, this is a good question, and is the same question that we asked to ourselves some weeks ago. Okay. So we are performing some experiment to understand if Sertoli cells reduce the transdifferentiation from myoblast to myofibroblast in a primary culture, or if the fibroblast that myofibroblast that usually occurs during the culturing of cells express less fibrogenic markers. So we are performing some experiment to understand if it's one or the other.
Okay. And maybe I missed it, but what you've done in vivo experiment, have you, so where you co-injected your Sertoli cells in vivo in muscle, do you see any reduction in fibrosis when you co-inject them?
Yes, yes,
okay.
We observe a reduction of adipose and fibrous tissues. Yes.
Okay, well, it's very interesting. And my last question, if I may, is you looked at DMD. Have you looked at other dystrophy where you might have fibrosis as well, or did you only check on DMD?
No.
Okay.
No, unfortunately, no. We are working just on Duchenne muscular dystrophy.
Okay, we might discuss later. Thank you very much.
I have a question too. It seems that the key factor is to maintain the in the myoblast the secretion of utrophin. Let me say, I expect that you interfere with the full differentiation of the myoblasts, or not? What is the timing of the?
I don't know if I understand well what you ask me, but probably I can explain this.
I repeat my question to do not lose time.
Yes.
In several of your slides,
yes,
you wrote the word utrophin, and yes, utrophin was usually increased
in myotubes. So we co-culture Sertoli cells with 4 days differentiation myotubes, and after 48 hours, we observed an upregulation of utrophin expression in myotubes.
But in mice, you have the same events, or yes, those are not correlated?
Yes, of course. After 3 weeks from a single injection in 4-week-old MDX mice, we have an upregulation of eutrophin expression in Sertoli cells muscle mice.
Yeah. Therefore, you are interfering with terminal differentiation of the myotubes, and this is beneficial. It is the same, let me say, all the project to avoid the evolution of the boys by
The upregulation
maintaining them as young as possible.
Yes,
And
exactly.
it is something like along this idea.
Yes.
I congratulate all the Sertoli. I can make in English the translation, because otherwise I will be misunderstood. And ciao Guglielmo, congratulations.
Hi Hugo, thank you all.
And we saved the 3 and almost 4 minutes, but I hope there are more questions for not specialists. I am not a specialist. I do usually stupid questions, but sometimes they are interesting. And no other people in the audience have
I'm
stupid suggestions?
Only a very simple question, maybe. What about the sustainability of such a kind of treatment? Do you know, is there a long-lasting effect, or do you have repeat the treatment every 4 weeks or maybe?
We have performed experiments for 6 months and also for 1 year. After 6 months, we have still beneficial effects of Sertoli cells. And we observed that after 1 year, Sertoli cells are still alive inside the peritoneal cavity of mice. So there are possibilities that they are still functional also 1 year, after 1 year.
Okay. Okay. Without any regression?
Without any regression.
Oh, okay.
For what we have seen for now.
Okay. This sounds promising. Okay, thank you very much.
And without any pharmacological immune suppression, of course.
Okay.